now let us discuss another two phytohormones, that is gibberellin and cytokinin. These two are also growth promoters. Okay, so starting with gibberellin. Gibberellin was discovered in Japan. Certain of Japanese farmers, they observed that certain rice seedlings would grow excessively tall and become weak and sterile. So this particular problem was then identified to be a disease, plant disease, and the disease was named as Wakani or foolish seedling disease. Okay. Later on, plant pathologist E. Kurosawa in 1926 found it to be a fungal disease that was caused by the fungi Gibberella fusicuroi. So from the name of the fungi itself, the active substance was termed as gibberellin later on. And because this substance is mildly acidic in nature, that's why also we call it to be gibberellic acid. Ga means gibberellic acid. Okay. Now, Yabuta later on extracted this active substance and called it gibberellin, okay, or gibberellic acid. Now, presently hundreds of gibberellic acids are known and are termed as GA1, GA2, GA3, etc. But you should know that out of all of these hundreds of gibberellic acid, the most important one, because it is the first to be discovered, is GA3, and hence it is also most extensively studied form of gibberellic acid. Okay. Now, the precursor to gibberellic acid is acetyl coenzyme A. Okay. And finally reaching to the functions of gibberellic acid. Now, gibberellin mainly is going to allow elongation of the stem elongation or elongation of the leaves or the fruit, that is going to be allowed by gibberellin. That's the major and the main function of gibberellin. So first is stem and leaf growth. It is going to allow lengthwise growth of the stem by allowing internodal growth, and also size of the leaf, size of the fruit. That way the plant will be grown under the influence of gibberellic acid. Next is bolting. Very, very important for your exam. That is bolting. Bolting means, see, in certain of roseate plants means plants in which during vegetative phase there is profuse leaf growth, but the stem elongation is going to be prohibited. Stem would not grow in height, rather there will be profuse growth of leaves. All these leaves, because they will arise from one single point, that's why the leaves will arise or leaves will appear to be arranged in the form of a flower. So this floral arrangement of leaf is known as roseate arrangement. Okay. So in roseate plants like cabbage, beet, etc., we see that in the vegetative growth phase there is no as such internodal growth that can be observed. Rather, all the leaves will remain arranged from a common point, right, in the vegetative phase. But just prior to reproductive phase, just before the beginning of the reproductive phase or just prior to flowering, in such plants we see that suddenly internodal growth will start and there will be a massive increase in the growth of length of the plant. Okay, so this sudden increase in the length of the stem of such roseate plants is brought about by gibberellic acid, and this phenomena is called bolting. Okay, so in roseate plants, sudden elongation in the stem length prior to flowering is called bolting. Alright. Now, next is seed germination. Seed germination is going to be allowed by gibberellin. It is promoted by gibberellin. How? Because when the seed has to germinate, there are various enzymes like amylase, protease, etc. They have to be activated because they will help in the remobilization of reserve food material. Okay, they will be, in short or in a simple way, if we have to place it, they will be allowing the conversion of complex carbohydrate into simple sugar that will allow the seed to germinate. And for conversion of complex carbohydrate into simple sugar, the enzymes which are required, that will be activated or remobilized by the help of gibberellin. Hence, gibberellin promotes seed germination. Do remember that. Okay. So these are the functions of gibberellic acid. That is naturally how gibberellic acid will be, affecting the physiology or the, functioning or growth or development of the plant. Now, finally, moving to the uses. That is, how can we commercially be benefited by the usage of gibberellin? First is fruit growth. I have already told you the lengthwise or size-wise growth of fruit or leaf is brought about by gibberellin. So it has been observed that commercially increasing the size of apples or bunch length of grapes can be brought about by the usage of gibberellin. Next is delayed ripening. Okay, ripening process can be delayed. You might be thinking that how can ripening, delaying the process of ripening be commercially beneficial? That's because if you can delay the process of ripening, the fruits will be there in the plant body for a longer time. Hence, you can increase the marketing period of the fruit. The fruits will then be available off season as well. And that's how they will be more costlier and they will be more commercially beneficial to the agriculturist, isn't it? So that way, by the usage, by spraying the plants with specific concentration of gibberellic acid, we can delay the process of fruit ripening, hence making them more commercially beneficial. Next is malt yield. Malt, I think all of you know, by fermentation of barley grains, malt is created and that will again be used in the brewing industry for further production of alcohol. Okay. Now, malt yield basically using the barley grains can be increased by the usage of gibberellin. Why? Because gibberellin enhances or promotes seed germination, isn't it? So, gibberellin will increase the sugar content in the seeds, and using such seeds will fermentation will occur, malt production will be increased. More the malt, more will be alcohol production. Okay. Next is sugarcane. I think all of you know that the sugar that we obtain is from sugarcane, and sugarcane, the plant, the edible part of sugarcane is nothing but the stem, and gibberellin promotes stem elongation, isn't it? So by the application of gibberellin, you can increase the cane length, and more the length, more will be sugar production. As much as 20 tons per hectare can be observed or can be enhanced by the usage of gibberellin. Next is quicker maturity. In certain of conifers, it has been found that we can make them, by the application of gibberellin, we can make them mature faster. We can quicken their maturity so that we can obtain the seeds in a much faster manner. Okay. So this is all about gibberellin. I think all of you have understood this. This is mainly a part which you have to memorize, which you have to mug up. So this chapter, the phytohormone part, is the most important part for your exam, and this part is nothing but for you to memorize. You have to remember these facts. Alright. So this is all about gibberellin. Now let's finally move to cytokinin. Now regarding cytokinin, discovery is about how F. Skoog and his co-workers, they had found that by the time when F. Skoog and his co-workers were acting on tobacco pit culture, they were acting on plant tissue culture method. So they found that in plant tissue culture method, when you have to perform the cell division in the petri dish, you have to add certain cell division promoting substances. So by then already auxine was known. Auxine had been discovered and it was known to promote cell division. But they found, F. Skoog and his co-workers, they found that when they were working with tobacco pit culture, just by adding auxine to the culture media, cell division rate would not be enhanced. Rather, apart from auxine or along with auxine, they also had to provide either coconut milk or yeast extract DNA to the culture media to finally to increase the rate of cell division. Okay, but what was exactly present in the coconut milk or what was present in the yeast extract DNA that was increasing the cell division rate was not found out by F. Skoog and his co-workers. Okay. So later on, Miller et al., he discovered that the first cytokinin was actually discovered by him, but it was a synthetic form of cytokinin, not naturally extracted from the plant. So what he discovered is that from degraded autoclaved herring sperm DNA, a cytokinin or cytokinesis promoting substance could be obtained, and it was chemically N6-furfuryl aminopurine, which was given the name kinetin. But kinetin, even if it was the first cytokinin known or discovered, it was not a naturally occurring cytokinin, rather it was synthetic one. Much later, finally, natural cytokinin was finally isolated from unripe maize kernel and was given the name zeatin. Okay, so naturally occurring cytokinin for the first time was obtained from unripe maize kernel and was termed as zeatin. Fine. Occurrence. Where exactly do we find cytokinin in the plant body? Cytokinin is found in any region of the plant body where cell division is occurring. Where continuous cell division occurs, there auxin and cytokinin both should be present. Okay, but in differential quantities. Fine. Precursor. Chemically, all the cytokinin are nothing but tRNA derivatives, and some important examples are zeatin, which is also commonly known as BAP, benzyl aminopurine, which is very much commercially utilized for plant tissue culture method. Okay, dihydrozeatin, IPA, isopentenyl adenine, etc. Fine. Now, moving to the functions of cytokinin. The function of cytokinin would be first, growth of leaves and shoot. Cytokinin promotes the growth of leaf, growth of shoot. It is basically going to allow cell division. So overall, it is going to have a positive impact on the overall growth and development of the plant body. Counteracting apical dominance. Now this is very important for your exam. You know that apical dominance means in the presence of apical bud, lateral buds would not be allowed to grow, and that is promoted by auxin, isn't it? So it is seen that even in the presence of auxin, the apical dominance can be prohibited if there is the presence of cytokinin present in the plant body. Okay, cytokinin, it will counteract apical dominance. Means in the presence of cytokinin, the auxin would not be able to stop the branching or the formation of the, or rather sprouting of the lateral buds. Okay, in the presence of cytokinin, even if auxin is present, the lateral buds will be allowed to grow. So that's how we say that cytokinin is counteracting apical dominance that was promoted by auxin. Okay. Now, delay in senescence. It has been observed basically by two scientists, Richmond and Lang, that cytokinin will delay the process of aging or senescence. Hence, we also call this particular part to be Richmond-Lang effect. Okay, that cytokinin, application of cytokinin can keep the plant parts younger or delay the process of aging. Cell division, that is the most obvious part by now. All of you know that cytokinin will be promoting cell division process just like auxin. Okay. Coming to the usage is first overcoming senescence, of course keeping a plant part much more younger, keeping it delaying the process of aging. That's going to be a very much commercially beneficial characteristics of cytokinin. Applying cytokinin to the plant parts can be making them more younger or can be delaying their senescence, and hence it is very much commercially important characteristics. Next, tissue culture, plant tissue culture. You know very well, plant tissue culture is a very important way, an advanced way how in very less of time a lot of copies or clones of plants can be obtained. A desired plant's clones, thousands of clones in very less of time can be obtained by using plant tissue culture. Now in plant tissue culture, auxin and cytokinin both are mandatorily required. If one is missing, plant tissue culture cannot be performed. Now, in plant tissue culture, there are three distinct phases. One is callus formation, in which we simply place the explant. That means the plant part from where you choose to extract the cells. You simply put the explant in the culture media where auxin is equal to cytokinin. When auxin is equal to cytokinin, the cells, which are usually permanent and differentiated cells, they will now become undifferentiated, and a mass of undifferentiated cells will be formed in no time, and that is what is known as callus. Okay. So after the callus is formed, eventually subculturing will be done, and then the cells will be placed onto a culture media where auxin is more than cytokinin. Auxin more than cytokinin will be promoting root formation, and eventually after this it will be placed to a culture media where cytokinin, if more than auxin, that will be promoting shoot formation. So first callus formation will be allowed by keeping auxin equal to cytokinin. Then actually shoot formation will occur where cytokinin is more than auxin, and then root formation will be allowed by keeping the shoots into a culture media where auxin is more than cytokinin. That's how the plant tissue culture is performed. Okay, so plant tissue culture is completely based on cytokinin as well as auxin. So that's all regarding these two phytohormones. Now next let us discuss about ethylene and abscisic acid.